It's day 3, so a few more days to go, and lots of lectures in the morning. And so now we move into the topics which are very relevant to what our program project is supposed to do, and where we focus on proteoglycans, glycosaminoglycans. So we're going to focus all of today, this morning, lectures on glycosaminoglycans. We have 3 special topics. We're going to focus on proteoglycan structures first, and then we move on into proteoglycan functions. Of course, it's very hard to just only talk about structures without talking about functions. So we can see some overlap of structures and functions, and then we're going to talk about various things associated with the biosynthetic pathways, what we know about the factors that control the biosynthetic pathways and catabolic pathways, and how they need to be balanced. If there's a defect in the biosynthetic pathway, what happens to humans, and what are the diseases that you can come across, and also defect in the metabolic pathways of this class of molecules also have severe consequences in humans. Number of genetic diseases are collectively called lysosomal storage disorders, and it causes a death in the early childhood, and it causes mental retardations and skeletal deformation. So we're also going to talk about the physiological, pathological functions. So then, towards the end, the third lecture, we're going to focus on how we can analyze glycan structures. By using HPLC, mass spectrometry, NMR, how we can fragment this molecule by using various techniques and characterize them. So that's why we decided to focus about first structures, and then functions, and then analysis of glycosaminoglycans. So let us start with proteoglycan structures. So proteoglycans, their home is extracellular matrix and cell surface. Okay, with the exception of one proteoglycan, cell glycine, which is found inside the mast cells. Almost all proteoglycans, with the exception of cell glycine, their location is on the extracellular matrix and on the cell surface. And so this proteoglycan occupies extracellular matrix. Extracellular matrix has a tremendous role to play in regulating cell functions, because it's like a gel. Extracellular matrix is like a gel where you have lots of collagen, and some of you might have done the tissue culture or organ culture, doing explant culture models where you have to use your matrix gels or some kind of gels, and those gel often contains proteoglycans, glycosaminoglycans, collagens, and so forth, because this extracellular matrix provides and has a specific role to play. First of all, it provides a mechanical support for giving cushion, and also it plays a role in differentiating cells and also triggering cell death through signaling pathways, or cell growth through signaling pathways, and also cell migration. Because when cells decide to migrate, or cells decide to divide and grow, and where it is going to go? It's not going to get smaller, it's going to have the same shape, size. So it's going to squeeze itself into the extracellular matrix. So extracellular matrix is the place the cells have to exploit their spaces. And so extracellular matrix play a role in regulating cell functions, and by providing signaling and sequestering proteoglycans too. And so if you look at the extracellular matrix, the major components is collagens, and also we have molecules such as fibronectins and also some other adhesive proteins. And that the major space of the extracellular matrix occupied by 2 molecules. One is proteoglycan, another one is hyaluronic acid. So hyaluronic acid is another glycosaminoglycan, which is a genetic molecule. So 2 molecules that occupy most of the space in the extracellular matrix, which is proteoglycan and hyaluronic acid. And so what is proteoglycan? So yesterday we talked about glycoproteins. Glycoproteins in which the carbohydrates are attached to proteins, and they have a prefix glycans. In glycoproteins, the major component of the glycoconjugate is a protein. If you take endothrombin 3, 45,000 Daltons. If you look at the amount of glycan, molecular weight is going to be 2,000, 3,000 Daltons. And whereas majority component of the glycoprotein is going to be protein. That's why the name is given as a glycoprotein. In proteoglycan, the major component is going to be glycan, in this case, glycosaminoglycan. So 90% of the mass is going to be provided by glycosaminoglycan. That's why now it just switched from prefix to suffix now. So in glycoprotein, glycans are the minor component, protein is a major component. In proteoglycan, most proteoglycans, and glycan is a major component, whereas a protein is a minor component, and that's why I switched the name from glycoprotein into proteoglycan. And this proteoglycan has a core protein, and to which you add the carbohydrate polysaccharide chains, and these side chains are called glycosaminoglycan because it has two repeating disaccharide units. One is hexosamine, one is uronic acid. It's called glycosaminoglycan. And this proteoglycan can exist in many forms, and either they can be anchored to in the cell surface via GPI. It could be GPI anchored, or it could be like transmembrane domain, like in the syndicans, and the transmembrane domain to which your protein is there and there are GAG chains attached to them, or it can simply get secreted like agricans, perlacans, decarin, and biglycan. All this proteoglycan gets secreted. So some proteoglycans are anchored through GPI anchor, and some proteoglycans are attached to cell membrane through transmembrane domain, and some other proteoglycans get secreted. And so all these proteoglycans have many different carbohydrate polysaccharide side chains, and heparan sulfate, chondroitin sulfate, dermatan sulfate, keratan sulfate. And of course, they also have a glycans, which we talked about, N-glycans, O-glycans. All these proteoglycans are also carry N-glycans, O-glycans, which we are not going to focus about it. And those N-glycans, O-glycans also provides a role for these proteoglycans to function. And of course, their molecular weight is going to be much smaller in these proteoglycans, N-glycans, O-glycan molecular weight. And also we have another molecule, which I just said a minute ago, hyaluronic acid. Hyaluronic acid is also glycosaminoglycan, but it is not attached to any core protein. And so these are the structures. If you look at this here, most of the proteoglycans, and they have 2, 3, or 4 or more glycosaminoglycan side chains attached to them, with the exception of few proteoglycans such as decarin. And majority of the proteoglycan have a 2, 3, 4, 5. In the case of aggrecan, they can have up to 100 chains attached to them. Okay, 100 chains attached to them. And each of the side chain can have up to hundreds of repeating disaccharide units. And if you look at the linkage region, because you have this proteoglycan, they're attached. These GAGs are attached to the core protein, and they're attached as specific residues, which is often serine residue, and to which what you do that you first add xylose. Xylose is a pentose sugar. You add xylose, and then you have a galactose, then you have a galactose, then you have a uronic acid, which is often, not often, is always glucuronic acid, and then you have hexosamine. This hexosamine could be galactosamine or glucosamine. So if you take proteoglycan and you look at the linkage region analysis, and you have protein and you have glycosaminoglycan side chain, but the GAG chains attached to core protein to serine amino acid through linkage oligosaccharide, a tetrasaccharide, and the tetrasaccharide always going to have the xylose, galactose, and uronic acid, and this could be hexosamine. Then you have a repeating disaccharide units attached to them. And in the case of hyaluronic acid, you don't have a linkage region because it is not attached to core protein. It's just a polysaccharide without any attachment to the core proteins. So without adding xylose group, you cannot make the proteoglycan. This is a starting point for you. And so even though hyaluronic acid itself doesn't attach covalently to any core protein like syndicans and glypican, it exists on its own, and it has a non-covalent interactions with proteoglycans. So this is aggrecan, which is secreted in the extracellular matrix, and they have a specific domain. The protein has a domain for hyaluronic acid binding. So what happens that, and so this domain binds to another small protein, and also it has another domain that can bind directly hyaluronic acid. And so that's how the hyaluronic acid, which is about 1 million Dalton, they can go all the way up to 1 million Dalton, 2 million Daltons molecular weight. Whereas the glycosaminoglycan side chains, which are attached to the core proteins like heparan sulfate, chondroitin sulfate, dermatan sulfate, keratan sulfate, their molecular weight maximum is going to be 60,000, 70,000, 80,000 Daltons. And maybe 100,000, which is you're pushing the limit. So most of the carbohydrate side chains, polysaccharide side chains attached to core protein like synthicans, glycans, the molecular weight is no more than 100,000 Daltons. Whereas the hyaluronic acid can have up to 1 million or 2 million Daltons molecular weight. And yes, Terry?
I need your office keys.
The coffee pot and everything is in our conference room on the top.
In the conference room?
That's what I did.
Oh, okay, because there's somebody in
There's
the
a
conference room. Okay, thank you.
And so they have a linkage region, protein domain that binds to both hyaluronic acid, and another small anchoring protein also facilitates interactions. And that's how when this proteoglycan, in particular glycan, gets secreted and gets excreted into cells, into the cells of outside, and then they would start giving gelation property. It's kind of cross-linking. Hyaluronic acid facilitates cross-linking all these proteoglycans, in particular glycan, and now it forms a big gel.
Do you know what the linker protein is, and is it always the same linker protein?
So the question is, do you know anything about this linker protein? It's always the same. And there are only about 2 or 3 known proteins, and which facilitates interactions between hyaluronic acid and this core protein. There are not many, just only 2 or 3. So when you look at the GAG types, which are summarized in this slide, so the hyaluronic acid, which is a non-sulfated glycosaminoglycan. This is the only glycosaminoglycan which is non-sulfated, and this is non-sulfated glycosaminoglycan which doesn't have any core protein attached to it, and it has repeating units of hexosamine and uranic acid, beta-1,4, beta-1,3 linkage, beta-1,4, beta-1,3 linkage. And so this is huge polymer. The case of chondroitin sulfate, which has a galactosamine and uranic acid, which is linked to beta-1,4, beta-1,3 linkage. And this chondroitin sulfate can be sulfated at the 2 position of hyaluronic acid or glucuronic acid occasionally. There are 4-sulfation, 6-sulfation of galactosamine. In the case of dermatan sulfate, which differs from chondroitin sulfate, and which has hyaluronic acid, chondroitin sulfate doesn't have hyaluronic acid, whereas dermatan sulfate has hyaluronic acid. When you have hyaluronic acid, they're most likely going to be 2-sulfated, not always, but you have increased tendency to form the 2-sulfation of hyaluronic acid. The heparan sulfate and heparin, which has repeating units of glucosamine and hyaluronic acid or glucuronic acid, which are sulfated at various places, N-sulfation, 6-sulfation, 2-sulfation, and so forth. Keratan sulfate, which is the one that we're not going to talk much. First of all, we don't know much about the keratan sulfate role and their biosynthetic pathways and catabolic pathways. And but nonetheless, this has the following repeating disaccharide units, in which what you have is a galactose, the galactose attached to glucosamine, beta-1,4 linkage, and this is glucosamine attached to galactose through beta-1,3 linkage. So in glycobiology community, and instead of writing names or drawing structures, and this is how the symbols are used, because this carbohydrate is very complex. They have alpha linkage, beta linkage, and they have 1,4, 1,3, 1,2, and 1,6, and then 2,3, 2,4, 2,6, and all different possible linkages. So in order to put the full structure possibly within the same page, and so that you can visualize it, and the symbol is often being encouraged to be utilized to represent the structures. So that's why we're able to put them all together in one single slide. If you want to put them all in a written structure. And also for biologies, the carbohydrate area is often dominated by biologies, and for them it's much more easy. Okay, it's easier to just put the circles and squares and then representing structures to visualize what they are. And whereas the chemists, they're very happy to push arrows and draw structures. We can put the Heparin ring, you can put the hexose ring, all different kind of conformations. And axial, equatorial, for biologists often not quite comfortable drawing structures with the axial, equatorial orientation and stereochemistry and so forth. Here, there is no stereochemical orientation you have to worry about. Just change the color. So the blue one is glucosamine, whereas the yellow one is a galactosamine. Of course, if you're colorblind, then you don't realize what the structures are, and that's why they're trying to be very careful, okay, when the community biology is involved in glycobiology research. They try to use the colors which are not linked to the colorblind people, and so that they can still facilitate seeing the structures nicely. So a lot of efforts went in, okay, and that's why I'm just trying to emphasize this. And it's critical. If you're going to use, talk, or you're going to write a paper and you're going to put the structures, and just don't choose your favorite color. Choose the color that has been recommended by the community through IPOC. So they have a specific nomenclature requirements, how you can use the symbols and what kind of color. And for example, glucuranic acid, and it has a blue color, okay, it's a diamond, divide by them, okay, 2, and the upper half you put the blue. Then it's inverted into iodinic acid, then you use this color, and then you put them on the bottom side the color. And so they would like to facilitate as much as possible for you to make it accessible for biologists and also for outsiders as well. So this is a chemical version of the molecule that you can see it, which I'm very comfortable with. And so we have a chondroitin sulfate here. So the difference between chondroitin and dermatan is simple. Chondroitin does not have iodinic acid, whereas dermatan has iodinic acid. And both chondroitin and dermatan, okay, has a galactosamine and glucuranic acid and iodinic acid, depending on dermatan or chondroitin. The beta-1,3 linkage and beta-1,4 linkage. And they can be 4-O-sulfated, 6-O-sulfated, and also could be 2-O-sulfated in the case of dermatan sulfate. And often you have a sulfation happens on the iodinic acid, but you can still have a 2-O-sulfation of glucuranic acid, which is a rare event, but it has been shown by Wolf, Lindahl, and others, in the very rare papers, that you have 2-O-sulfated glucuranic acid found abundantly during development and also in the brain, in the central nervous system. You have 2-O-sulfated glucuranic acid found only in central nervous system and during development. 2-O-sulfated glucuranic acid is found. But otherwise, most of the cases, you only see 2-O-sulfated iodinic acid, not the 2-O-sulfated glucuranic acid. And as the names are, okay, the chondroitin sulfate was originally isolated from chondrocytes. So they gave the name chondroitin, origin from chondrocytes, originated from chondrocytes, they gave the name chondroitin. And dermatan sulfate, it is not dermarine, it's a dermatan, and it was isolated from derma cells, which is the skin cells, and that's where you see large amount of this iodinic acid containing chondroitin sulfate, B, which is called dermatan sulfate. And heparan sulfate, heparin, and if you look at the stereochemistry, here you have a galactosamine, that here you have glucosamine, and so glucosamine linked to glucuranic acid or iodinic acid. The skibulin means it can have two different stereochemical orientation, could be gluco or iodo, that's why I put the skibulin. So if you look very carefully, and there are two differences you can see in chondroitin versus heparan. One is the linkage. Here you have beta-1,3 linkage, beta-1,4 linkage. But here you have beta-1,4 linkage and you have alpha linkage. Dr. Desai talked about alpha and beta orientations. So chondroitin has only beta linkages, whereas heparan sulfate has both beta linkage and alpha linkage. So beta-1,4 and alpha-1,3 linkage. And between chondroitin and heparan sulfate, chondroitin has galactosamine, in which hydroxyl group, the fourth person is up. Whereas in heparan sulfate and heparin, it is a glucosamine, where the hydroxyl group is equatorial. Of course, it is not the hydroxyl, it's like ether linkage, in which you have this oxygen, hydroxyl oxygen is linked to the C1 carbon of the uranic acid. So it's an ether linkage, glyceric linkage it forms. So it has a beta-1,4 and beta-alpha-1,4 linkage. And it has glucosamine but not galactosamine as a repeating units. And both heparan sulfate and heparin name came from the fact that when someone isolated this molecule from hepatic tissue originally, then it's given name heparin. And so that's how the name is given for heparin. It was originally first time isolated from hepatic tissue. So origin of hepatic tissue is given heparin. And but both heparan sulfate and heparin are somewhat similar, but you will see more in detail about what the difference between heparan sulfate and heparin later. And the keratan sulfate has a repeating units of galactose attached to N-acetyl glucosamine, which has a beta-1,4 and beta-1,3 linkage. So unlike other glycans, such as chondroitin sulfate, dimethyl sulfate, heparan sulfate, heparin. Keratan sulfate has only hexosamine, in this case glucosamine, but does not have a uranic acid. So keratan sulfate does not have any uranic acid, it has a galactose. So it's not galacturonic acid, it's just a galactose. And so chondroitin, dimethyl, and heparin, they all have a carboxylic functional group. Whereas keratan sulfate does not have a carboxylic group, okay, in their polysaccharide chain. So galactose with glucosamine, it's a neutral series, okay. Of course, the sulfated are 6th position of galactose and the 6th position of the glucosamine. Is it clear? So if I ask you to write at the end of the day, you'll be able to write the structure, at least symbol. So I know this is very hard, and so unless you keep when reading and practicing yourself, and the practice really helps you. And also keep in mind that whenever you draw the structure, and make sure that this link, this carbon, you can see this one, and this one, try to put them parallel. I mean, this one should be parallel to this, and this is parallel to this one. I think sometimes people put OH like this. You should not put the OH coming this way, okay? This should be all parallel, of course slight, okay, but slanted, but it's parallel. Like that, you can see this and this oriented, okay, aligned. And this link is also aligned. And like that, this aligned and this aligned. So they're all, okay, nicely parallel. So don't draw structures with the amine group coming like this, or OH coming like this OH coming down like this. And so then sometimes it misrepresents structures, and also the orientation is different. So you have to follow this way of orienting everything, this way, this way, this way, this way. So then it's easier for you to also understand what they are. So these GAG chains are very complex, and they bind to a number of different proteins, and they have a number of different roles to play. And the chondroitin sulfate structures are shown here. Now it binds to many different proteins, plays a role as an anticoagulant, and plays a role in forming our bones, and also facilitate neurite growth or inhibit neurite growth. And it also binds to a number of adhesive proteins, and also binds to signaling molecules, morphogens, Wnt, okay, like FGFs and other BMP and so forth, and it can regulate a number of different functions during development and beyond development. And dimethyl sulfate has been shown to bind to heparin cofactor 2 more specifically, and then it can function as an anticoagulant, which is a different mechanism than the heparin function as an anticoagulant, which happens through antithrombin 3 binding. So dimethyl sulfate functions as an anticoagulant to binding to heparin cofactor 2. And also dimethyl sulfate has been shown to bind to hepatocyte growth factor, HGF. And in the case of keratan sulfate, it is not well understood, first of all, and except the structures, okay, and with the exception of structures, and we don't understand much about their biosynthetic pathways, and we don't understand completely what are the factors that interact with them. And but nevertheless, it is known that the keratan sulfate plays a role in maintaining transparency of cornea. That's why I can visualize, I can see you all. And also masking the immunogenicity of agarcan. With agarcan, which I just said in the very first slide, the structure in which you have a cold protein with hundreds of side chains attached to it, chondroitin sulfate. They also have, between the chondroitin sulfate attachment sites, you also have keratan sulfate, which is much smaller in size compared to heparan sulfate, chondroitin sulfate. But you have like 10, 20, 30, 40 different number of chains of keratan sulfate attached to it. And it has been suggested that by having this keratan sulfate, it can mask the immunogenicity of certain proteins and regions. Like when this is the G1 domain of agarcan. Agarcan has various domains, and the G1 domain of agarcan has been suggested to be masked, which may be immunogenic in humans, even though we make it ourselves, but still could be immunogenic. But if you remove that keratan sulfate, maybe we may have induced immune response against our own molecular resynthesis. So maybe there could be evolutionary reason why you need to mask that G1 domain with having keratan sulfate there. And with respect to heparan sulfate, we know a lot of things about heparan sulfate and heparin. They've been shown to bind to FGF1, FGF2, and also other FGFs, hepatocyte growth factors. We know a lot of more things about how it functions as an anticoagulant by binding to antithrombin 3. It has also been shown that it binds to GD, glycoprotein D, of the herpes simplex virus, and thereby it facilitates the entry of herpes simplex virus. And also we know that this heparan sulfate plays a role in HIV infection by binding to TAT protein TAT, and so forth. And we know a lot more about heparan sulfate heparin and still there are a lot of unknown things. So the biological functions of GAGs often facilitated through not just by itself, by binding to the growth factors and proteins and thereby modulating signaling pathways, resulting in regulating, as I said many times, differentiation, migration, growth, embryogenesis, organogenesis, left-right axis inductions, and wound healing, and so forth, some of which are described here. And also heparin molecule and dimethyl sulfate or chondroitin sulfate has been shown to bind to LDL, and this LDL is a lipase basically, which cleaves the cholesterol and removes the cholesterol and the bad lipids from our body. So this has a lipase activity, and by cleaving off the lipids from circulating the blood, and it can clear the lipids from our body and cholesterol from our body, and it plays a major role. And this lipase cannot function without the help of heparan sulfate. This heparan sulfate, proteoglycan, anchored to the endothelial cells on the surface, and then on the endothelial cells facing the blood flow directions, and there the LDL lipoprotein lipase attached to and anchored to the heparan sulfate, and then looking out for these LDL particles as is going through the blood vessels, and then they'll hit the LDL particles and then start cleaving and breaking them down, and then taking them back into endocytosis pathway. So the lipid maintenance in the body is controlled by lipoprotein lipase, and whose activity depending on anchoring itself to the heparan sulfate, which is attached on cell surface of endothelial cells. So now let us start focusing on the biosynthesis. We talked about structures, we made different proteoglycans. Let us focus now on each class of molecule. Let us start with the chondroitin sulfate, and you have same amino acid. And the first thing you do, you add xylose, then galactose, then you add glucuronic acid, and then the galactosamine is transferred with the help of GalNAc transferase 1. So now you add the GalNAc here. So then there's an enzyme called chondroitin synthase. The chondroitin synthase is a bifunctional enzyme. Bifunctional enzyme, I mean it can do two different things, not a single thing it can do. So the chondroitin synthase is a bifunctional enzyme. It has two different transferase activity, glucuronic acid transferase activity and GalNAc transferase activity. So therefore it can transfer both glucuronic acid from UDP glucuronic acid and galactosamine, N-acetyl galactosamine from GalNAc UDP, and resulting in having repeating disaccharide units attached. So chondroitin synthase is a polymerase enzyme. It adds alternatively galactosamine and uronic acid. And it doesn't add only galactosamine, it has to always add both galactosamine and uronic acid back and forth. It has two domains. One domain adds the galactosamine, and then glucuronic acid is added, then galactosamine is added, glucuronic acid added. So no one knows the exact mechanism as how this happens, this bifunctionalism adding repeatedly two different units. So some speculated that you assemble, pre-assemble galactosamine and uronic acid, you attach them, galactosamine, uronic acid, you attach them. Some suggested that you add galactosamine, then you have to wait for some time, you add uronic acid, then you add galactosamine, uronic acid, and it is not quite understood exactly what's the mechanism, but nevertheless you have to add both galactosamine, uronic acid alternating manner, so that you end up having repeating disaccharide units. And again, it is not well understood in the field what controls the chain length, because I just gave you the synthase can make this polysaccharide, repeat disaccharide N. N could be 20, 30, 40, 50, and has up to maximum 100 repeated disaccharide units. But what controls the number of disaccharide attached to the growing polysaccharide chain? It is not understood well. So one could speculate maybe the precursors, the UDP glucuronic acid and UDP galactosamine can control, and also maybe some other unknown factors can regulate the action of the synthase and thereby regulating the chain length. And the next enzyme is glucuronic acid C5 epimerase. Again, in chondroitin sulfate, it's not quite clear what is the sequence of events that results in forming this 4-O-sulfated, 6-O-sulfated, 2-O-sulfated repeating disaccharide units. And because in HS is going to be different, you can see that. And even though the slide here shows that you do C5 epimerase first and then 4-O-sulfation, but a number of other people suggested that, and both C5 epimerase and 4-O-sulfotransferase has to work together, because a 4-O-sulfation of this galactosamine, 4-sulfation of galactosamine actually facilitates conversion of uronic acid into iduronic acid. Of course, some other group says that, well, you can do this, convert gluconic acid to iduronic acid, but it has not been completely understood whether it happens first and then next this happens, or you do 4-sulfation and uronic acid formation in parallel, okay? And this parallel is not quite understood. And then you have 6-sulfation and the 2-sulfation happens, resulting in forming this complex structure. So there are a number of proteoglycans which are exclusively committed for chondroitin sulfate formation. Of course, one is agarcan, and so it's a core protein, and which can have up to 100 polysaccharide chondroitin sulfate attached to them. They're secreted proteoglycan, and they're found in cartilage, and that avoids the friction when we run and walk. And as we get older, we make less agarcan. As you make less agarcan, what happens? This has a gelation property. It's a huge molecule with hundreds of chains attached to them. Each one has 100 repeating disaccharide unit. You can have a 50,000 Dalton chain, each chain. When you have 100 chain attached, think about that. 100 by 100,000. How many? Molecular weight. How much? 10 million? 1 million? 100 times 100,000. 10 million. Yeah, then, okay, when you have 10 million, okay, then this 10 million Dalton agarcan can attach to hyaluronic acid. Hyaluronic acid is itself 1 or 2 million Daltons molecular weight, and in each hyaluronic acid, you can put 50, 60, 70, 80 agarcan attached, anchor non-covalently. It's a complex. So now you multiply this by 100, 10 million by 100. It's like, becomes a billion, okay, molecular weight. So it has a gigantic, okay, means molecule, so holds large amount of water, and that's why we avoid the friction between the two joints when we walk and run. But as we get older, and enzymes going down, their activity is going down, their expression is going down, and also the precursors production is going down. So what happens is that you make less agarcan, less truncated, less sulfated. As yesterday Dr. Desai said, which I didn't know until yesterday, that each sulfate, I know they can bind to water molecule, but they can bind to 9 water molecule. So think about that. You take the 1 billion Dalton non-covalent, okay, and covalent complex, you multiply this by different number of sulfate you have, times 10, times each sulfate groups, then it becomes so huge. That's why it's able to hold water molecule. But then you can squeeze them, the water is out, but as soon as you take the pressure out, the water goes back inside. And so that's why as we get older, then the mechanical damage happens and we have more pain. Then we take glucosamine, or we can take orange juice with chondroitin sulfate. I don't know how much benefit it gives, but I know many people who take orange juice with chondroitin sulfate, they feel good. And that's evidence that it works. And so the Vesican is another proteoglycan with 12 to 15 chondroitin sulfate attached to it. And even though the agarcan is secreted in the cartilage, is also expressed in brain. And both cartilage and brain, okay, it holds large amount of water molecule. You can't have a brain dehydrated. And the brain is placed because it's very soft tissue, large amount of water is hold, and again, the agarcan helps to hold the water molecules in the brain. And of course, it has more than just holding the water molecule. And also, agarcan has been shown to facilitate the chondrocytes formation and differentiation during the early development. And then you have Neurocan, Brevican, and SLRP is a very interesting molecule, and so it stands for small leucine-rich proteoglycan. The 2 proteoglycans comes under SLRP. One is a decarin, one is biglycan, and you can see that they have only 1 or 2 chondroitin sulfate chain attached to them. They're secreted and found in the connective tissues. And other examples are shown here. And the phosphacan is one, I think, that Vimal in my lab is interested, and that might probably play a role in the action, guidance, and growth and degenerations that are expressed in the brain. Also, a number of other proteoglycan exist, and also cell glycine, which I just said, which is produced only in the mast cells, but is not secreted, is inside the cell. And so that has not only the chondroitin sulfate cell glycine, also has a heparin attached to it. So this slide shows the structures of the proteoglycans, examples of proteoglycan, how the chondroitin sulfate proteoglycan, how they look like. You can see the agarcan, a lot of different structures attached to it, and they all anchor to the surface. And with the exception of phosphacan, and when it's a short version, it's a long version, which is secreted out. And of course, agarcan also secreted out, but they can bind to hyaluronic acid. So, but the chondroitin sulfate has many different sulfation pattern. They're called A, B, C, D, E. I also know they also call F, G, and then they have hyphen. They put superscript, subscript, I, and okay, lot of different names. But A, B, C, D, E are the most abundantly expressed, are well-studied molecules. And chondroitin sulfate A basically has a 4-O-sulfation. Chondroitin sulfate B has hydronic acid with and without 2-O-sulfation, and chondroitin sulfate C has 6-O-sulfation. So if I ask you chondroitin sulfate A and C difference, is that chondroitin sulfate A has a 4-O-sulfation, C has 6-O-sulfation. Chondroitin sulfate B, the another name for the chondroitin sulfate B is a dermatan sulfate. In the olden days it's called chondroitin sulfate B, but in modern days no one calls this as a chondroitin sulfate B. Now we call as dermatan sulfate. But dermatan sulfate is the same as chondroitin sulfate B. If you read literature in 1980s and 90s, they say chondroitin sulfate B, but the modern literature says dermatan sulfate. But both are said, don't get confused. What is chondroitin sulfate B and dermatan sulfate? There's no difference in terms of structures. And chondroitin sulfate B is what used before, but now what we use is dermatan sulfate. And so chondroitin sulfate D has both 2-O-sulfation of either glucuronic acid, hydronic acid, and 6-sulfation of galactosamine. Chondroitin sulfate E has 4,6-disulfate disaccharide. And these molecules again expressed in cartilage tissue cells and also in brain, and they found secretory granules. And so, but this source is given here because these are the sources from when you get these molecules from commercial sources. And so they also said where they obtained from. The shark has a large amount of chondroitin sulfate D. So that's when the company produces this and sells you for the research. And they go for the source, major source where they can get this large amount, and then they can purify, they can sell it to you. So these are sources from which the isolated being sold by Sigagago and other companies. So the sulfation of chondroitin sulfate, okay, influences dramatically and signaling pathways in action, guidance, and growth, and some of which is listed here. What are the factors involved, and is given in detail here. And I don't want to go through in detail, but you can just go through when you have time. There's a lot of different specific molecules, factors involved, but they play a major role in relating the action, guidance, and growth. Now let's move on into keratan sulfate, Proteoglycan. Even though it's not well understood and studied molecule, and so structures of the keratan sulfate molecule is given, and they're all often secreted, some of which is bound, is given here, and they're found in two places predominantly. One is in agarcan. You can see the keratan sulfate attached in just opposed to the chondroitin sulfate in agarcan, and they're also found in specific proteoglycans shown here. They're expressed in the central nervous system and in cornea. And unlike other proteoglycans like heparan sulfate, chondroitin sulfate, dermatan sulfate, and this molecule is so very unique in the sense that yesterday we talked about what is this one? Linkage. What kind of glycan is this? N-glycan, fantastic. Asparagine you have. So galactosamine, you have glucosamine. Glucosamine is a chitobiose. Then you have mannose, so trimannose chitobiose. You have fucose attached to it here. And then this N-glycan, when it reaches here, and so now you have, at some point down the road now, you have this galactose and glucosamine repeatedly added to make this keratan sulfate. In the case of chondroitin sulfate, dermatan sulfate, heparan sulfate, you have xylose attached to core protein, then galactose, glucuronic acid. Whereas in keratan sulfate, it is xylose has no role, it's an N-glycan, and to which you add them, okay? You attach the N-glycan first to the core protein, then one of the arms will be utilized to make keratan sulfate chain. In some cases, it even said that maybe 2 arms are utilized, but majority of the cases you have a single arm attached to it. And also, this one is a sialic acid. The sialic acid caps this arm, and also one paper, if I recall correctly, it also caps right here. Even though it is not shown here, it can also cap the sialic acid has always been shown to occupy the terminal position of many glycoconjugates. Likewise, here also the sialic acid occupies this one. Of course, it's not extended, and also the sialic acid can cap this keratan sulfate here. And there are two different kinds, KS1 and KS2. And KS1 is asparagine-linked N-glycans, and this one is a serine threonine. So what this kind of glycan is? So O-glycan. So the O-glycan also you can add them, but here also is capped with this one, and also is likely might be capped here as well, but I need to go back and check. But if you recall correctly, one paper that talked about capping this place as well. So what is the function of keratan sulfate? They will be shown to play a role in maintaining transparency in the corneas. And also another function for this keratan sulfate is that it gives like a shock absorber in Glycan, because Glycan is the one that's constantly bound, undergoes mechanical stress all the time because the cartilage is exposed. Also, this keratan sulfate provides some kind of shock absorbent properties. And how this molecule is synthesized, it is not well understood. And so most of our knowledge of how the biosynthesis occurs derived not from animal models, not from in vivo studies, but often derived from cell culture models. You take a variety of different cells, you can genetically manipulate, you can overexpress the enzyme, you can downregulate the enzymes, you can add the molecules to inhibit, like tunicamycin to affect the N-glycans and so forth. And so it gives you enormous ability to like rapidly find out the factors that can control and chain length. You can manipulate so easily, but manipulating the animal models, each one may take like several years and so forth, knocking out things. Of course, now we have gene knockout and gene transgenic mice models available, but again they're very expensive and time consuming, and the cell culture model often utilized to advance our knowledge about the factors that control the biosynthesis, mechanism of biosynthetic pathways by many groups. And in the case of keratan sulfate, why we are not able to go further is because you take a cell, the keratan sulfate expressing cells like corneal cells and keratinocytes where you have a keratan sulfate is expressed. As soon as you isolate these cells from animal models and trying to culture them in vitro system, the cells are fine, but they don't express keratan sulfate. They don't express keratan sulfate in cell culture. People tried for more than 25-30 years and they gave up. Okay? And you, as soon as isolate from animal models, you try to grow them in cell culture model. They don't express keratan sulfate. So maybe something is there in the animal system, okay, animal model, that can facilitate formation of keratan sulfate proteoglycan. But once isolate the cell that expresses keratan sulfate proteoglycan, try to grow them in vitro, it's gone. They don't express. Cells don't die, but they don't express keratan sulfate. So when they express keratan sulfate, then you can start asking questions about the factors that control what happens, all those things. You know, many of the enzymes have been cloned, okay, understood well about that. The cell culture model is very difficult to be utilized and as a model to understand the factors that control the biosynthetic pathway, which is the state of the knowledge right now for the keratan sulfate. Let's move on to heparan sulfate now, and which our lab focuses on. And the very first enzyme that acts upon this core protein is xylosol transferase, which transfers the xylose from xylosol UDP into this core protein, which is the single most important step in the proteoglycan biosynthetic pathway, because once you add the xylose, that core protein committed to make proteoglycan. If you don't add xylose, you don't have proteoglycan produced. So xylosylation is a very first step. I don't say single most important step, is a very first step and rate limiting step, and that commit itself to converting the core protein into a proteoglycan. If you don't have a xylosylation, you don't have proteoglycan production. So xylosol transferase, which facilitates transfer of xylose from xylosol UDP into core protein, that is the first step committing itself to make the proteoglycan. And then you add the galactose. Galactose is a galactose beta 1,3. I know that the group from Pittsburgh and Maryland and Harvard working with a gal alpha 1,3 gal knockout pig models to understand how we can use the pig organs for transplanting in humans. That's a gal alpha 1,3 gal linkage. This one is the galactose beta 1,3 galactose linkage. Keep in mind, so don't get confused. The linkage has a big difference between beta and alpha. But they have beta 1,3 here. There is in pigs they have a gal alpha 1,3 gal linkage. Now here, but they express gal alpha 1,3 gal epitopes. But here's a gal beta 1,3 gal. So different enzymes involved in making this linkage region than the one that expresses gal alpha 1,3 gal. Then you have glucuronic acid added, and then in the case of chondroitin sulfate, you add glycosamine as a fifth residue, 1, 2, 3, 4, 5. Whereas in heparan sulfate, the fifth residue is glucosamine. So here's the fifth residue, 1, 2, 3, 4, 5 is glucosamine. So still we don't understand quite exactly what controls this linkage region and to commit itself to make a heparan sulfate chain or chondroitin sulfate chain. But all we know, if the fifth residue is a galactosamine, is going to become chondroitin sulfate, no matter If you add the fifth residue of galactosamine, it's going to become a chondroitin sulfate. If you add the glucosamine, it's going to become heparan sulfate. It looks like it's a magic catening point. You add the galactosamine, it becomes chondroitin sulfate. You add the glucosamine, it becomes heparan sulfate. That means, and there's a competition going on, the winner is the one that controls it, or there is no competition. Actually, it's a factor that regulates how you add the galactosamine or glucosamine. Also, what we haven't shown in this slide, which you should be aware of, which is not shown here, both this xylose, this galactose can have a phosphorylation. Xylose can have a 2O phosphorylation, the xylose, and the 2% can have a phosphate, and this galactose can have a sulfate as 6 position and 4 position, 6 position and 4 position. So you can have a galactose residue on a 6O sulfation, 4O sulfation, and also xylose can have a 2O phosphorylation. And all those things perhaps can play a role in controlling chain length and controlling whether you add galactosamine or glucosamine as a fifth residue, which then subsequently dictates whether it's going to become HS chain or CS chain. But current knowledge is we don't understand. It's a belief in the field that it controls it, but exactly no one has shown how this controls it. Unlike this, sulfation happens in galactose and galactose residue, and the phosphorylation of xylose is a transient event. So phosphorylation happens in xylose, and then the chains are made, but at some point down the road, the phosphate also is cleaved off. So the phosphorylation is not that you add the phosphate, it sticks there forever. The phosphate comes off at some point down the road during the HS biosynthesis. So it's transiently phosphorylated and then it's cleaved off. So there's an enzyme involved in phosphorylation and there's an enzyme involved in removing it. But not always a transient. In some cases, the phosphate is found, but in most of the cases, phosphate is removed. So is either transiently attached and then removed, in some cases attached forever. And again, maybe the transient attachment and removal are like permanently adding the phosphate group could also control the expression of specific HS and CS and chain length. Again, we don't understand why this phosphorylation happened and what is its role still. Yes?
Can you have the synthesis of heparan sulfate and the other one in the same protein, on the same protein?
Yeah, so the question is, on the same protein, can you add both heparan sulfate, chondroitin sulfate? Yes. In some core proteins, we can add both heparan sulfate and chondroitin sulfate. For example, Syndican has both heparan sulfate and chondroitin sulfate. If we go back to the very first slide. See, you have Syndican, for example, here. It has HS chains and the CS chains are shown here. So Syndican have both HS and CS chains. Whereas the Glycan has only HS chains. Agarcan has, even though it's not shown here, it has chondroitin sulfate and also it has a Keratan sulfate. Of course, Keratan sulfate is not as big as chondroitin sulfate, but it also has Keratan sulfate, but it's not shown here. But it has both chondroitin and Keratan sulfate. And then Perlacan has only HS, and then Decarin has. So some Proteoglycan has both HS and CS, and Agarcan has both CS and KS.
But are they position specific?
The question is, are they position specific? Yes.
According to
Yes.
protein amino acid.
Yes. The question is whether the Syndican here is shown, you have HS chains here and CS chains here. They are specific? Yes. You cannot put the HS chains here, and you cannot put the CS chains here. You always put HS chains here, you always put the CS chains here.
So that itself also dictates whether the growing thing is going to be a CS or a HS.
Yes. So one thing I haven't talked about it, so he's saying about the attachment sites are highly conserved, that's true, which also dictates whether you're going to have HS chain or CS chain. That's very true. And also it has been shown by Jeff Esco a number of years ago with Lujan Zhang when she was a graduate student. Phenomenal paper was published in 2 or 3 papers are shown. They made a synthetic peptides in which they put serglycin. So whenever you have the serine residue where you add the GAG attachment sites, when you have multiple serglycin, multiple, not one serglycin, whenever you have multiple serglycin, and that facilitates the formation of GAG chains. And also by changing amino acids and having tryptophan and tyrosine and few of the hydrophobic amino acids, they've shown that it can facilitate formation of heparan sulfate, but not exclusively heparan sulfate, but predominantly heparan sulfate, maybe 60%, 65% heparan sulfate, 35% chondroitin sulfate. But that's a peptide they used it to determine whether they can undergo GAG formation, and also what type of GAG is predominantly produced on those attachment sites. So they found that more cell glycine you have repeating amino acid cell glycine, it has a greater probability to become a GAG chain attachment sites. And also by having hydrophobic residues, certain residues, it can facilitate the formation of heparan sulfate, and maybe 60%, not how many percent, maybe 60%, 70%. But if you take the same sequences in the HS here, and you have mostly HS is formed. But if you look at the sequence differences, this specific attachment site and this attachment site is not great. But yet, your HS is always attached here and C is always attached here. That means there is more than just say a sequence, like a primary sequence. There's a ternary structures involved maybe, and also maybe other modifications happening here but not here, maybe here but not there, on the amino acid sequences, maybe end glycosylations, old glycosylation, which are also found in syndicans, glycans everywhere. And those old glycosylation, end glycosylation may control the folding and accessibility of the xylosol transferases and subsequent enzyme synthesis. Again, this is all unknown. And but what is known right now is that some cold proteins can have both HS and CS, and some cold proteins can have only CS and CS and KS, or only CS, and so forth. But the attachment sites are highly conserved. It's not that you take this decarin, one day here, one day here, one day here. It's not like this. You take the decarin, and no matter what the tissue is, and attachment site is always conserved. You can take different tissues, different cells, different organisms, okay? But the decarin attachment site is always conserved, and same thing with syndicans and glycans. This is so amazing, phenomenal. You take different species, attachment site is always conserved. Of course, the structures are different in many different cells. I'm talking about structure means GAG side chains, sulfation pattern different, domain structures are different, but the attachment site is conserved. The linkage, the chain length is not conserved, but attachment site is conserved. For example, Syndican, again, another phenomenal paper from Ralph Sanderson when he was postdocing with Burnfield, another giant in the field, like 1970s and 80s. And they have looked Syndicans and expression in lung development, and the mouse like they call E9, E10, E11, like different embryonic days, post-embryonic, what they found that syndicans during development, lung-specific epithelial cells, and the chain length is so different. And at some point, one of the attachment sites have a longer chain, and sometimes they have a shorter chain. Then again, they switch back to longer chain. So there's a polymorphisms, what they call and coin the name, syndican exhibits polymorphism such that the same cell during development, and they have different chain lengths, and still is not understood why. And so maybe by controlling the chain length, you can control what kind of factors they can bind, and hence the signaling, and so forth, what they're supposed to become eventually. But the attachment site is conserved. So we talked about, so now there's various enzymes involved. The very first enzyme is NDST, which we have shown that recently known as is the first enzyme. And people always thought it's a gateway modification. And so next is the C5 polymerase and 2-OST acts upon, which converts glucuronic acid into iduronate acid, as shown here. And the 2-sulfation of iduronate acid happens. Of course, 2-sulfation of glucuronic acid is also shown here. That can also happen, which is much more rare than the 2-sulfation of iduronate acid. And then you have 6-sulfation happens, and then you have 3-sulfation, which thought to be a rare modification, which still proves the rare modification, at least in adults. And so then that confers some properties such as anticoagulant properties and herpes simplex viral infection in cells and animal models shown. So 3-OST is a rare modification, but nevertheless results in forming a very complex molecule with variations in chain length and sulfation pattern. But one notable difference between heparan sulfate and chondroitin sulfate is that chondroitin sulfate does not have domain structures, like there are sulfations everywhere, whereas in heparan sulfate you have regions which are heavily sulfated and you have regions which are non-sulfated. They call NA region. NA means N-acetylglucosamine, and non-sulfated is called NA regions or NA domain, whereas NS domain, which stands for N-sulfated glucosamine domain, which are heavily sulfated. As you can see that there are a lot of small circles, which stands for sulfate. You can see it's a heavily sulfated epimerized NS domain. You also have interdomain between NAS, NS and NA domain, which is called NA-NS domain. You have number of gluconate residues, and you also have some N-sulfated residues, which are epimerized and thiosulfated. So you have an NA region, non-sulfated region, and you have sulfated NS region. You have also a mixed domain called NA-NS region. And why? In chondroitin sulfate, you don't have such kind of things, like, but they are sulfated randomly everywhere. But here in heparan sulfate, even though the sulfation seems to be random in NS region, but they are in a specific place. But you also have a number of residues which basically produced without any modifications. Why? Still, we don't understand why we have domains like NA and NS. But we know that having NA domain is very critical for protein binding, because once you start doing sulfation, probably people in Dr. Desai's lab and Krishna's lab done a lot of modeling studies, and when you start doing more and more sulfate, the molecule becomes very rigid, and the helical, they become more rigid. But when they're non-sulfated, they're more flexible. They can just bend around. And that's why hyaluronic acid is used as a long structure where it can attach the agar-can. They can bend around, and provide the mechanical so they can bend. When you make a gel which is very rigid, then it's not good. You want them to bend around, So the molecule can undergo the mechanical stress they can bend. That can do it because hyaluronic acid is not only big, it's non-sulfated. Whenever you add sulfate to the molecule, it becomes increasingly rigid. Add more and more sulfate. So perhaps one can predict that by having non-sulfated region and sulfated region, as a protein, chemokines, growth factors binds, is going to not only change the conformation of the protein, it's also going to change the conformation of the carbohydrate as well, so that they can get the right conformation, right biological activity to trigger. And when to do that, there should be some place some flexibility exists. They all become so rigid, you can't do anything, and that's not good. So by having these regions which are very rigid and where the binding takes place believed to be, and then having this kind of non-NA regions or NANS region which are more flexible, perhaps you can facilitate binding of multiple growth factors, morphogens, and chemokines to facilitate the signaling pathways to be triggered. And that again, it has to be demonstrated by having only NS region on a syndican to show that it doesn't have any more functions, and or to have only, of course, by having only NA regions, you won't have any functions. You need a sulfation, but no one has made a syndican with highly sulfate NS region only to show it has a function still or not have the functions. But I believe that NA region is means everyone knows that NA regions is more flexible. Therefore, maybe that facilitates. Maybe make it more efficient for syndicans to function as the receptors to regulate the signaling pathways.
Are these enzymes specific to heparin sulfate, or they can sulfate the chondroitin sulfate also?
Okay, the question is the enzyme that I talked here about, NDST, C5-ephemeris 2, 6-3-OST, they're specific for heparan sulfate. There are a number of enzymes we talked about before for chondroitin sulfate. They're specific for chondroitin sulfate. And these enzymes specific for chondroitin sulfate, dermatan sulfate, and these enzymes specific for heparan sulfate. As far as I know, and I think some people also report there's no cross-reactivity. So they are specific for HS, they're specific for CS. And another big difference between HS and CS is that in CS, there is no N-sulfation, whereas in HS, you have N-sulfation happens.
And one more, does these enzymes follow the sequence pattern what you wrote, like first N-sulfation, then 2-OST, or it can do like randomly any sulfation?
Okay, the question is whether enzymes, as shown here, follow the sequence of reactions, or they can jump around. And so the sequence of reactions shown, just for simplicity, to understand that N-sulfation happens, then epimerization happens, 2-sulfation, 6-7-3-O. But N-sulfation is still required. Without N-sulfation, you cannot epimerize uronic acid into iodinic acid. Okay? Without N-sulfation, you cannot convert glucuronic acid into iodinic acid. And again, the 2-sulfation, 2-OST enzyme, prefers iodinic acid more than glucuronic acid. Okay? And also 6-OST enzyme works more efficiently on N-sulfated region than the non-sulfated region. And also, it is known that to some extent from Wolf and Bass' work that the 2O sulfation cannot happen after 6O sulfation, and the 3O sulfation has proved always lost because this enzyme that was rare modification was cloned. Okay, of course, all 3OST happens to be cloned first before all other enzymes was cloned, and but it was pushed at last because it has a specific biological functions. But this is how the field right now represents it, and we also shown NDST reaction can happen after all this modification happens. You can still bring the NDST reaction. Here, you can do N-sulfation that we have shown in a recent paper. And even though this slide gives you a sequence of event that happens, but the sequence is not rigid, but you have to follow certain rules that you cannot do safe epimerization without N-sulfation. And the 2O sulfation is preferred aromanic acid, but it also can happen in gluconic acid occasionally. So those are the rules you have to keep in mind that governs the reactions. But what I believe is happening is that, and as polymerization is happening, even though we show the polymerization is finished and then you do this reaction, so polymerization is happening, like elongation is going on, and this biosynthetic enzyme start acting on this place, and so as it's acting on this place, elongation continues, then somehow the enzymes can jump from here to here and can carry out some reactions, but some of the enzymes that cannot jump can still do these reactions depending on how the enzymes can go from one part of the structure into another part of the structure, can grab and hold, do the modifications, and that could eventually dictate generation of NA, MS regions and MS regions, and still we don't understand. But the chain as undergoes elongations, and one place you may doing N-sulfation, epimerization is happening, another place you can do 6O sulfation, another place like maybe is more matured, so maybe the region which is more close to the linkage region is more matured, and because it's what made yearly, and so that maybe more modifications happening, whereas the region that's still elongating is going on, the growing front, maybe the less modification happened because my enzyme has to move from here to there.
So these are parallel, like once the...
Yes, exactly. They are parallel. These are these reactions happening on the same chain, multiple places in parallel. It's thought that is not proven yet, and because it's very hard to also prove. It's so complex, as you can see, so many reactions happening, and this all happened within half an hour or so forth, because this turnover is 2 hours for this Proteoglycan. Many other Proteoglycan can have a turnover rate of 2 hours, is made within half an hour and secreted in the cell surface or extracellular matrix, and when cell surface bound, they have life of 2 hours maximum, and they'll just suck back inside the lysosomes, is broken down. So they're made rapidly and displayed rapidly, and then the life also made rapidly, and but they have roles probably by controlling the turnover. And so this reactions is happening in parallel and many different places.
So you suppose you want only 6-sulfation or something on the chain. So how efficient the reaction is, like without other sulfations?
You're talking about in vivo or in vitro?
In vitro.
In vitro. And without other modification, you only want
You
to...
cannot control, right?
Excuse me?
In vivo you cannot control having a just 6O sulfation.
You can control in vivo by knocking down enzymes. And gene knockout experiments, you can do it. But of course, in vitro is very easy. You can take enzyme that you want and use it to modify it. You can do it.
So how efficient it is without having remaining all the sulfations? Like
you can do that, but what we found that N-acetyl group, is not preferred for 6-OSTs. N-sulfation means it's really good for the 6-sulfation. N-sulfation seems to be facilitating 6-sulfation. But if you just take non-sulfated one, you can still do the sulfation with the enzymes, but they're less efficiently. Again, keep in mind, these molecules are made in given cell, maybe picogram. Your one cell may be making femtogram. That's all it makes. And so if we take a million cells, we get like maximum 5 to 10 microgram. If we take 10 million cells in T75 flask, the maximum we isolate 10 microgram, and that's all pretty much we isolate. From 10 million cells, 1 to 10 million cells in T75 flask, we get maximum 10 microgram. If you do a math on one cell, you perhaps get like femtogram, that's all. Yet their biological roles, it's a small quantity made.
Back to your slide on the heparin, so the NS and NA domains, do you see any kind of a pattern as a function of chain length? How do they repeat, like the NA and the NS? It's like 10 disaccharides or like 20, like does it depend on the chain length?
Okay.
Do you see any kind of
Yeah. So the question is whether the NA-NS domains, the chain length of NA and NS domain is conserved. The answer is no. And even the chain length itself is so different during the development. And so there are no specific pattern that we can generalize. NA, okay, you're going to always have this NA region, and there's always going to be NS region, and there is no generalization you can just draw from what is known from the literatures, and there are no generalizations you can make. But predominantly what is known that NS region happens to occur more towards the end of the chain, and whereas more towards the core protein, it seems to be more NA region. They're less modified, more towards the core protein, and more towards the terminal of the chain, more modifications happening. One can assume perhaps that core protein is there, maybe the enzymes cannot access so easily as the chain is extended, maybe the enzymes can more easily access, but that means the enzyme has to wait until the chain length is grown this much. And so again, that's a mystery in the field and it's not well understood. But what is known that NS domains can more predominantly found towards chain terminal, and NA domains are found more predominant towards xylose, like more towards the linkage region. That much I can tell. But what is happening in between and how much is conserved is not conserved.
So maybe that is due to the fact that the enzymes has to work, so the initial domain should be so flexible so that if it is rigid, all the things cannot happen.
Yes.
You need flexibility so that this can come and work.
Yes. But not on the chondroitin sulfate though. Yes.
Generally, the sulfation pattern is specific for the proteins like FGF1 or chemokine. Is this unique for heparin? That means the sulfation pattern.
So sulfation pattern unique for the proteins or unique for the core proteins?
Generally, the sulfation pattern is specific to a specific protein like FGF1 or chemokine.
Okay.
Probably the specific sulfation pattern identifies a specific protein like FGF1 signaling or EGF signaling and some growth factor and chemokines also.
Yes.
Is this unique or sulfation pattern is for different things?
So for some proteins, people have shown that the sulfation pattern requirements for binding to the growth factors and specific proteins of interest is unique. And what is emerging now is that in some cases is unique and some cases it's not unique. There is a promiscuity involved, or more than one structure can have the same protein they can bind, they can facilitate maybe the same function or maybe a different function. So binding doesn't necessarily mean that it has a biological function.
And it's unique, how can it recognize the different proteins like that? Probably there should be some difference, the sulfation pattern.
The binding of the protein to sulfation pattern depending on the protein structure. That is not controlled by the HS chains. The binding of the protein to the specific sulfation domain depending on how the basic amino acid residues or the other factors that control the topology of the protein, and that would dictate whether they can bind and recognize a specific sulfation pattern or not. So maybe I'm not understanding your question exactly.
So, can I answer? So Krishna Mohan, I don't know whether you realize that if this is a template-less synthesis, right? There is no template. And that really means that whenever the enzymes come over and they recognize a certain sequence, they would put in a sulfate. So sometimes some chains will have a sulfate, some chains will not have a sulfate. So what typically happens is that you get millions of sequences. So in those millions of sequences, you may have few sequences that recognize a growth factor. Some other sequences may recognize antithrombin. Some other sequence may recognize IL-8. Okay? Now, for most part, what he's trying to say is that there are very, very few, as of now, there are very, very few proteins that are really known, for which we know that this is the sequence of heparan sulfate, which is absolutely needed to convert, to generate a biological response.
Yeah, for the FGF family, we know FGF1 and FGF2 family.
That's a big question mark.
Yes, still like even though people said, even though people have...
Think that, oh, FGF is a specific interaction. But it's a big question mark.
So I think more towards end of this lecture, I'm going to also show, people have shown that FGF receptor complex formation in 6-sulfation, and we have shown, Tao has shown in a paper published like a few months ago, that without 6-sulfation they can still form a ternary complex. Ternary complex, ternary complex is more critical for signaling than your binary complex. And people always say the FGF receptors and FGF complex needs a 6-sulfation.
So FGF induce the signaling.
Yeah. So, but Tao's paper has shown that it forms a ternary complex without 6-sulfation. So how do we explain that? So there's still a lot of unknowns, and so we have to look systematically. It's possible that everybody is right, but if they write their own science when they're looking at their own cell types, at their own model systems, and maybe, okay, that's how it is. Maybe all of those things are... It could be cell type dependent, and also like the same protein expressed in many different cells with a different glycosylation pattern, hence different conformations, and hence it's possibly right that same proteins, okay, but they express different cell types of different conformation, and then they can bind to different structures of HS sequences. But at the end, it not necessarily have the same results. They can have different functions. Different binding may result in different extent of signaling, different level of intensity of signaling, and hence you don't want to have a continuous firing of the signaling, and maybe you want to have a moderate signaling, maybe by slightly different conformations, different topology that recognizes, maybe the K on and off rates, not I'm talking about in vitro system, biological system binding and can releasing themselves, could trigger how much signaling you want to keep triggered and how much you don't want to trigger. And so that will have huge, we have a continuous on signal, it's not good.
Do you have any specific enzymes inside the cells that can desulfate, like specifically sulfate?
Yeah. So the number of enzymes involved, I think that as you move along, we can see the number of enzymes involved. Specific enzymes clips off, and this 6-sulfation removed by 6-sulfatase, and N-sulfation is removed by sulfamidase, and 2-sulfation removed by 2-sulfatase. So absence of any of those enzymes results in accumulation of these molecules in the Golgi and causes lysosomal storage disorders, and results in death, early childhood death, or mental retardations, and so forth. So there are specific sulfatases which removes the sulfate groups, like a sulfotransferases which add the sulfate groups. The sulfatases which clip off the sulfate groups from these molecules, which are localized in the lysosome. So sulfotransferases which are localized in the Golgi, sulfatases are the enzyme which clip off the sulfate groups, are localized in the lysosome, and they remove the specific sulfate groups.
So that can answer the question that they can specifically give a heparin sulfate needed for a protein with different sulfations. Like he's asking, like some sulfation is not needed for a protein to bind or something, right?
Yes.
So these sulfatases can do that and make a chain which doesn't have a 6-O-sulfation which is sufficient for the protein to bind.
Yeah, but keep in mind, this sulfotransferases the sulfation in the Golgi, right after that they get secreted in the cell surface. Only when they finish their lifetime, they titrate to the lysosome to undergo desulfation. So those lysosomal resident sulfatases have no role to play in desulfating. But there's other enzyme exists called endosulfatase, and which we call sulf1 and sulf2, and which are known to specifically remove the 6-O-sulfate groups, and which are secreted in the extracellular matrix. So when in response to some signaling, it's possible that sulf1, sulf2 with sulfatases which get secreted, and they can come and do the 6-O desulfation, and thereby can trigger unregulating signaling how much you want them to bind. And again, in some disease conditions, sulfatase enzymes are secreted. They're supposed not to be secreted outside, maybe only during early development they get secreted. But also in cancer cells, also they get excessively secreted, and in adult tissues they should not get secreted, only during early embryonic development. And in cancer cells, the sulfatases get secreted. They specifically remove 6-O-sulfate groups. Endo-6-sulfatase means it can remove the sulfate groups within the chain. Whereas the one that I talked about, the specific sulfatase removes specific sulfate groups, which can only remove from terminal residues, exosulfatase, desulfating enzymes. Whereas the endosulfatase can do endo-desulfation, that means can bind to chains, internally desulfate, and those are the ones are known to regulate cell signaling, growth factors binding. Yes, Spencer.
That the same protein core can have both GAG side chains and N-glycosylations. So what's the mechanism of control for whether N-acetylglucosamine is added to a serine residue or whether xylose is added? So like whether that same residue can have either type of glycosylation?
So the question is, okay, the serine residue undergoes xylosylation, and the serine residue also can undergo galactose modification, then that forms O-glycans. So what controls whether this one is going to be O-glycan or this is going to be a glycosaminoglycan attachment site? And perhaps the answer lies on the cell glycan repeating units. When it is there, maybe that site is then stored and rescued or reserved for attaching the GAG chains. And if there is not many repeating cell glycine, maybe that sites can undergo and become O-glycans sites. And also keep in mind, xylosylation is not happening in Golgi. This is happening in endoplasmic reticulum as the protein is made. Maybe, as you know, there's a question yesterday from Krishna, which happens first. N-glycosylation is your co-translation modification, whereas the O-glycan is a post-translation modifications. Likewise, glycosaminoglycan elongation modification is a post-translation modifications, okay? You keep adding the chains and elongating and adding sulfations. Whereas xylosylation is, I believe, is not your post-translation modification. I believe, I don't know, okay? But it has been shown xylosylation happens in pre-Golgi. That means it happens in endoplasmic reticulum en route into the Golgi. It has not exactly been shown which place is happening xylosylation. It happens, all that has been known is a pre-Golgi. It could be endoplasmic reticulum or en route from endoplasmic reticulum into Golgi in the vesicles, the xylosylation is happening. So that means that there won't be any competition with the other, O-glycosylation. Of course, there could be competition between N-glycosylation and O-glycosylation, but O-glycosylation, xylosylation happens only on serine residue, whereas asparagine residue undergoes N-glycosylation, okay? And therefore, there is not enough. It's possible that whenever you have N-glycosylation, and that's a site may not undergo O-glycosylation, because I said yesterday, your asparagine X serine, and that's a site can undergo most likely N-glycosylation. So when you have such a huge N-glycan attached to it, maybe you cannot add xylose, you cannot extend the glycan formations. Maybe that the sites which they are not N-glycosylated nearby, by having cell glycine repeating units, maybe it prevents the N-glycosylation happening, because whenever asparagine is most likely can undergo N-glycosylation. So by having cell glycine multiple repeating units, the nature ensures that there's a site reserved for glycan attachment site. You don't want to have N-glycans attached there, because N-glycosylation is a co-translational modifications. So that is the case, if we add the xylose next to N-glycan, maybe the subsequent enzymes involved in making galactose, uranic acid, though it may not be able to access that site. So maybe by having repeating cell glycine, maybe nature engineered in such a way that that site is reserved for GAG attachment site, by preventing N-glycosylation happening, and which could provide a steric hindrance to the enzymes.
Generally, proteoglycans exist in the extracellular membrane. How these trans across this membrane?
Excuse me?
Where did synthesis happen? Like
Which one?
proteoglycans.
Yes.
It exists in the extracellular membrane.
On cell surface as well.
Cell surface only, synthesis only. Synthesis also.
Excuse me?
How these will trans across the membrane?
Okay, so the question is, so this molecule undergo all these modifications with the core protein attached to it, and so how this molecule get outside the cell? So they get secreted, because most of the Golgi is a very mosaic, very flexible, vesicles. So what happens is, it's not a vesicle, it's like, but then they get secreted in the vesicle forms. So because Golgi is the one can be constantly undergo dramatic changes, and there's a cis Golgi, medial Golgi, and trans Golgi. So most of the proteoglycan has to go through cis to medial to trans Golgi. From trans Golgi, and they can bud out like small vesicles. Also, it has been shown the cis Golgi as well can release the vesicles as well. And so then that can go to the trans Golgi. From trans, they can come back to cis. But at some point, the vesicles, once the maturation happens, the vesicles will just take them all into the secretory vesicles, and they have their own domains and sequences and signaling, and that facilitate and not going to lysosomes, not go to the nucleus, not go to other compartments or mitochondria. They facilitate the secretion by having secretory signals into the membranes. They anchor from inside and then just get fused from inside and then just get released. If it's going to be no transmembrane domain, it just gets secreted in the extracellular matrix. If they are the membrane anchored domain, then it's just anchored by a GPI or just transmembrane domain. So there has to be the vesicles before they just leave from Golgi. Okay? Still, it's not clear? So this slide basically talks about, okay, domain structures, and you can see the NS domain, you can bind. What?
Give them a break.
Okay. So we'll take a break, then we'll come back.
But I think, you all really raised absolutely interesting questions, and most of I think the answers to many of your questions are not fully defined. Okay? I mean, this is such an open field that a lot of work will need to be done in order to really say whether there is going to be a defined size length for NA, NS-NA, and NS domains, as to how the sulfation occurs. A lot of things are not clear. So, I mean, that's one of the reasons why, they think that glycoscience needs to be heavily supported. So, this is the right time to be in the field.
Yes.
These stands across the membrane, like that the cell itself is carrying some proteoglycans.
Yes,
sulfate. It continuously dissociate and continuously produce and transfer to the membrane.
Yes.
This is continuous process.
Yes.
So,
Because the gold itself is not
it's
it's very transient. The gold is very transient, right? And the dramatic and changes all the time. So some of them, like it's a budding out, continuously budding out. And so some of the secretory vesicles will take not only the proteoglycans, it also takes other molecules as well, other proteins, collagens, they all have to secrete it out. Right?
it depends on cell type.
Yes.
Some cancer cells is
No. Because all cells have to get secreted in the mouth or attached to cell surface. They need to be vesicles. Yes. The molecule itself cannot transport itself from one place to another place. It has to go in a vesicle. Because molecule cannot transfer themselves from one place to another compartment or place within the cell. It's impossible.
Actually I worked on the FGF1 non-cluster pathway. FGF1 non-cluster pathway.
Okay.
In 6 years I worked on the FGF1 protein complex.
Okay.
It forms multi-protein complex and then comes out of the membrane during the stick conditions.
So whom do you work with?
Chinyu in Taiwan.
In Taiwan. Okay.
Yeah, actually we solved this protein complex structure. So FGF1 this one on a protein and with this method.
But with HS chain, did you ever something?
No, without HS. Later we focused on the FGF1 drug design. I worked with the disaccharides.
Okay, disaccharide synthesis or
No.
like interaction?
One of the collaborators there.
Yeah. Okay, I see.
We worked at SOS and small molecules.
Yeah, no stars. Right. Good. Yes, next one.
Could you explain? So I've been doing a lot of reading on this because of the lecture that you gave in MedChem. But I'm confused as far as the linkages. So I know that when you say alpha or beta linkage, I know what that is as far as the reducing sugar end. But I don't understand this. So
We're only on the third slide now to go this much.
yeah, several pages. I think there's like 9.
This is only my part of the slide. Shalaja has to give a talk.
Oh wow.
I don't know what is going to happen.
I'm gonna
Okay.
take. But okay. So I guess it's 2 questions. So here, oxygen usually has a bent stereochemistry. So then I understand that if you have a sugar here, this
Yeah.
is alpha linkage.
Yes.
And then here, I don't understand how you do this. So then this means that you have this type of linkage, so you have this sugar,
Yeah.
right?
Yes.
How does this work? This one?
What does that means?
Yeah, I don't know what that means. So
Yeah, this is like basically the older nomenclature they use it.
Okay.
And the right way of doing it, okay, I would recommend So you see one of the slides that we had?
Yeah, so it's
So
this one.
don't go like this way. And so you put a straight line and straight line is better.
Okay,
Don't
so
blend
that
it.
means that it's below the plane.
Yeah, it's below the plane. And this is above the plane.
So this is above the plane for this one.
Yeah.
But below the plane?
Yes. Exactly. Because it's not, the molecule is bent like this, okay? Means even though we put it like this, in 3 dimensions, the molecule is like this. So for them, this is above. For this is below. And it goes like this, structure. For example, the ring is like this. Don't think the ring is like this. Okay? Think the ring is like this. So for them, this is above. For this is below. You understand? See, this is the ring. Right? This oxygen is above. And this is below the ring. So you put the ring like this. Yeah, staggered. Not quite staggered. Staggered is like this, but this is like this. So now you can see what I'm saying? But when you write them in the 2D field, you have to put it like this. So you have to put it like this.
I understand that. And then, so when you do, so we were talking about beta 1,3 linkages. I don't understand where your third carbon is. So this is your anomeric.
Yeah.
Is it linked to here?
Yeah, yeah, yes, yes.
Okay.
So what's happening is that this is beta 1,4, right? And this is beta. It goes like this. And here this goes like this.
Oh, okay.
Okay? Beta 1,4, beta 1,4, beta 1,4. But we just always go like this. And this is a beta 1,4. That is a beta 1,4. That means another 6. Okay? So I'll drink water and come back.
Beyond that,
Yeah, there's some, like most restaurants don't. And restaurants, I just saying there's sauce. But I have it now, that applies.
Okay. Is it okay? Is there? I'm asking you. I mean, this is slightly different. It is on, right?
It is on.
Okay. Okay, good.
The only thing is that sometimes at the moment we're just
Okay, so we start. Everyone is back now? No? Miss? Okay. Let's just get going though, because I think I covered only 1/3, and so I haven't even printed for the structure analysis yet, and so because the papers ran out, so I need to go to the office and get the papers, then I need to print it and then bring them back. So this is only 1/3 of what I covered so far, then I need to also print few, more. So these are the examples of the heparan sulfate proteoglycans, like perlecan, agrine, and syndicans, beta-glycan, glycans. So the major family that has been extensively studied by many groups is syndicans 1, 2, 3, 4, which are isoforms, and also glycans 1, 2, 3, 4, 5. Actually, they have glycan 6 as well, if I recall correctly. And so here the coating prods and the number of chains attached is shown. And so these are mostly like specific for the HS, but also syndicans have chondroitin sulfate attachment sites, and also the glycans, the beta-glycan has one HS and one CS as well. So this slide depicts and shows how the proteoglycan looks like. HSPG specific one, the syndicans, and the glycans one is the GPA anchored. The syndican ones are the transmembrane protein anchored, and then you have a few other, CD44, which is non-natural. I think they call CD44 proteoglycan as, if I recall correctly, is transient proteoglycan. It is not always having GAG chain attached to the CD44. It's not always attached to them. It's a transient. That's why it's called as a transient, a temporary proteoglycan. And we don't understand why. Sometimes you have a GAG chain attached to it. Sometimes it's not attached to it. And that still comes under heparan sulfate proteoglycan category. And then what are the functions of proteoglycan? So as we studied, they play a role in anticoagulations, role in enzymatic, action guidance, embryogenesis, also regulate inflammation, and action growth and guidance and degenerations. Also, of course, viral infections are normal functions, but various infectious agents also can bind to HS, and they can affect. And all of these things, the function of proteoglycan exercise, they exhibit these various functions through binding to specific proteins, either from the virus or from pathogens or other proteins of interest in a specific biological system, and that results in having this function. There was a nice review article published by Esco's group like a few years ago, and it talks in detail about physiological roles of HS, and then by covering the last 50-60 years of work. And it's a very nice review article written and focusing extensively on HSPG's physiological functions in vascular systems, renal, lung, heart. You can just read. It's a very nice article, so read that when you have time. And so basically it exercises its function through binding to various proteins, and they're very critical for many various physiological processes by binding to specific proteins which are listed here. And the one that would be interesting for Krishna's group and the chemokines for anticoagulations, Dr. Desai's group is interested in looking at antithrombin 3 and heparin cofactor. And I don't think much is known about protein C factor interaction with HSCS, if you believe correctly. But there are some papers that describe both the and CS in particular role in protein C inhibitor activation. So this is a model being proposed. Many different models are proposed in the field of both HS field and also FGF. Even though HS regulates various functions, they're binding to FGF. We have two different, completely different group of scientists. There are people who focus only on FGF, they don't care anything about HS. There are people focusing on HS, they don't care about FGF, but they work with FGF and to show the importance of HS. And the FGF group, uses HS to show the FGF importance, and they do crystal studies and so forth. And also within FGF community, there's many different models being proposed, and you have two FGF receptor and one HS chain, or two HS chains binds are called 2-to-2-to-2 complex, or 2-to-2-to-1 complex, and some other group proposed that 1-to-1-to-1 complex, one HS chain binds to one FGF and one FGF receptor. And I think most of this, okay, the hypothesis and model proposed based on the crystal studies, and also the proteins are expressed most of the time in E. coli system, and also keep in mind that most of the crystal studies carried out using commercially available heparin. Heparin, as you all know, has heavily sulfated, whereas heparan sulfate is much less sulfated, which is coming in the next few slides. And so we can come to the conclusions one way or other is possible that for some of the growth factors, 2-to-2-to-1 model maybe is right. For some of the growth factors, 2-to-2-to-2 model is right. For some growth factor receptor interactions, maybe 1-to-1-to-1 model is right. Maybe in some, okay, maybe you don't need even growth factor. For example, FGF4 can trigger the signaling without even receptor, and so that could be 1-to-1 complex. So, but all our theories that exist in the field, I'm just bringing this to you, but I'm not inclining towards one way or other complex. And so you're aware of the complexity involved in the field, so that as you make progress in your research, to keep these things in your mind, to integrate what is known in the field and what is unknown, so that you can integrate your hypothesis with whether it fits with one model or other. In the case of anticoagulant action of heparan sulfate is fantastically well defined through competition between Bob Rosenberg's lab and also Wolf Lindahl's group in Sweden. Exactly what are the sequence requirements, what are the specific sulfation patterns required in order to confer the anticoagulant activity, and of course it doesn't involve about the kinetic aspects of that. That's what Dr. Desai later on shown 15 years later, the specific sulfation and how much it contributes to the binding component of the antithrombin 3 that results in activating factor or inactivating factor 10A, and so that's a different aspects of the focus from Dr. Desai's group and published very nicely in JBC. But nevertheless, the pentasaccharide sequence shown, okay, with the different color code is pentasaccharide. It's not penta, it's actually hexa, right? But is the color code is a small segment of the polysaccharide. Upon binding to antithrombin 3, it changes the conformation of antithrombin 3, which facilitates the binding to factor 10A. As soon as it forms the ternary complex, antithrombin, factor 10A, and the pentasaccharide comes off. So as soon as the factor 10A binds to antithrombin 3, the pentasaccharide comes off, and so that it can go back and bind to another antithrombin 3. So in essence, this pentasaccharide carbohydrate functions like a catalyst. So enzyme by definition, okay, is a catalyst, right? So it used in a small quantity can do multiple reactions again and again and again. Likewise, this heparin is like a catalyst in inactivating factor 10A by binding to antithrombin 3, but therefore it accelerates by 1,000-fold by having a specific sequence. But antithrombin itself can inactivate factor 10A, but it's very slowly is done. By using this pentasaccharide, it can accelerate by 1,000-fold, and inactivation rate is accelerated really very fast. And then by capturing factor 10A, and now it forms a reversible complex, and basically that factor 10A is not available, which basically converts the prothrombin into thrombin, and the activated thrombin now converts fibrinogen into fibrin that forms a clot eventually. So basically by capturing and sequestering factor 10A and by forming the complex irreversibly with antithrombin 3, and you prevent the conversion of prothrombin to thrombin, which eventually converts the fibrinogen to fibrin that causes the clot. So that's how this pentasaccharide molecule functions like an anticoagulant. So because this forms a coagulation, this is a coagulation cascade, and by withdrawing factor 10A from the coagulation cascade, you slow down dramatically this rate of converting prothrombin to thrombin, and therefore that you decrease the clot formation, and that's how it functions like an anticoagulant then being used in surgical procedures. So what is this? You all know about heparin and heparan sulfate, and outside they always get confused. What is the difference between heparin and heparan sulfate? So heparan sulfate is expressed by all cell types, and they're expressed in the extracellular matrix, and they're expressed also on cell surface, whereas the heparin is produced only by mast cells in the connective tissue, and they are produced and stored inside the mast cell. They are not get secreted. Even now it is not well understood what is the real biological function of heparin. We make this heparin in our own mast cells but stored inside the mast cells. But what is the biological function of heparin? which is still not well understood. So people have knocked down serglycin, core protein, in which you add the heparin chains, and also knocked down the enzymes which are specific for heparin, and to look out for the specific phenotype. Surprisingly, the mouse survives without making heparin in the mouse cells. Although the mouse cells don't have a heparin in the knockout mice, but the mice cells survive. So then what is the physiological role of heparin is not well understood. But nevertheless, and we know that heparin can be used as an anticoagulant by deriving this from cows and pigs and whatnot, animal sources. And so there are specific differences exist between heparan sulfate and heparin besides their origin and where they're stored. And heparin is, in terms of size, okay, it's about maximum 28,000 Daltons and between 7,000 to 28,000 Daltons. And so unfractionated heparin, uncleaved original endogenously produced heparin stored in as a serglycin proteoglycan in the mouse cells, it has a maximum of 28, even though it says 20, it has 28 kilodaltons maximum weight. And of course, when you want to use as an anticoagulant agent, you cleave them with peroxides, bleaching agents, and also with enzymes and sodium hydroxide and various techniques and like irradiation with a specific wavelength of heat, and then you cleave them into ozonylysis, fragment them into smaller sizes, and you sell them as a low molecular weight heparin. Because of heparan sulfate, they can go up to 70,000-80,000 kilodaltons, but not more than that. Also, that if you look at heparan sulfate, they have a domain-like structures, non-sulfated region and sulfated region, and also they have a less sulfate density, I would say. Of course, N-sulfate is less, and they have a lot more N-sulfate. And if you look at hyaluronic acid content, it never exceeds, even 50% is very high. I would say like 30% or 40% is what maximum you would see. And whereas in the case of heparin, they have 70%, 80% hyaluronic acid, greater than 70% hyaluronic acid in heparin. Because heparan sulfate is a glucuronic acid is a major component always. In the case of heparin, hyaluronic acid is a major component. Of course, the antithrombin binding to the heparan sulfate is very, very low because less sulfated. The probability of generating a specific sequence that binds to antithrombin is very low. In the case of heparin, 1/3 of the heparin, unfractionated heparin, binds to antithrombin 3. And 1/3 of the fractions of the unfractionated heparin can bind to antithrombin 3. And it's basically synthesis by all cells, whereas heparin is made only in the connective tissue mouse cells. So when you when you read your own paper, don't get confused with heparin and heparan sulfate, because I've seen people always outside the field mix both heparin and heparan sulfate just randomly. And one line they say heparan sulfate, we studied heparan sulfate. Another line said that heparin is okay, we cleave them into these molecules. And so they use heparin and heparan sulfate inadvertently. They think they are all same, but they are not same. Okay, heparin is different, heparan sulfate is different. But they are similar molecules, but they are not the same molecules. So this heparin that we all use as an anticoagulant is derived from animals, and there are a lot of problems involved. Originally, until 1990s, it was isolated from cow and used. Of course, cow is big compared to pig. And so after 1990s, and because of the thanks to British people and mad cow disease, and people are scared, and so then what happened that until 1990s, everybody used the bovine heparin as an anticoagulant at hospitals everywhere, all over the world. So after mad cow disease, and because of US FDA strict regulations, and they don't want to use anything in humans, and that can cause mad cow disease. Like they don't want to absolutely any issues with that. So then what happened that they switched from bovine source into porcine source sometime in 1990s. Then the problem is that in US, a lot of people here, we eat beef, and not a problem. So a lot of poultry from which you can get the intestines, lung, and bovine lung in particular. And also the cow is big, so you can get a lot of heparin, no problem. But once we switch from bovine into porcine, even though we eat here porcine, the pig we eat, not that much, and because pig is smaller, and we have less population here, we have only 250 million people, and it's not a big, the porcine source. Then what happened that eventually, when we switch from bovine source into porcine, and then much of the manufacturing of heparin is switched into pigs naturally because of mad cow disease, and then we switched also manufacturing from US into third world countries, into China in particular, and that's a major source of production of heparin. And but keep in mind is that it has specific problems of any time when you use heparin from animal source. One is that it's from animal origin. Animal has its own pathogens and infections and so forth, and it can cause cross reactions with the humans. And also keep in mind it is not coming from one pig, okay? It's coming from 600 million pigs. Think about that. How complex, the molecule itself is very complex. Variations exist from the same pig and so many variations. When you mix up 600 million pigs and get this heparin in tons and tons, thousands of kilograms for world supply, so how much heterogeneity exists? So, and also with the recent problem of adulteration of heparin with chondroitin sulfate, which is somewhat a similar structure sulfated polysaccharide, but which does not have an anticoagulant activity, chondroitin sulfate, via antithrombin 3. It can function through cofactor 2. But if you oversulfate chondroitin sulfate chemically, if you oversulfate chemically, not enzymatically, chemically oversulfate chondroitin sulfate, then it starts binding to antithrombin 3. So it binds to antithrombin 3, then it has an anticoagulant property. Then that's great. Why? Because from any given pig or cow, when you isolate heparin, you may get X amount of heparin. I don't remember exact amount, okay? But chondroitin sulfate is always made 20 times, 30 times, 50 times more than heparin, even in humans, okay? And chondroitin sulfate is made 20 times, 30 times, 40 times, 50 times more than the heparin. So obviously, when you sacrifice a cow and pig to isolate from a given tissue heparin, like you have 50 times more molecule chondroitin sulfate, which you don't use it, and we sell it at a cheaper price. But once you oversulfate, it has the same activity as a heparin. So then obviously then you can start making more money, 50 times more money than just only selling heparin. So there's intensive chemical adulteration that happened 2-3 years ago that results in causing death about 80 or 90 people in the US and also worldwide. People died. So that says a potential problem with animal derived heparin, whether it's intensive adulteration or accidental or the indigenous problem of using animal heparin. Because believe me, this is the only molecule that is used as a drug for more than 80, 90 years actually from animal source without any very homogeneous form. It's highly heterogeneous. And so the solution to this one is chemical synthesis. The problem is that it's very challenging, and also you can use enzymatic synthesis, which is also equally challenging. You need so many different enzymes, and at the end of the day, what can get you to the market is how cost effective it is and how scalable it is. And right now, the enzymatic synthesis is now started rising above the bar, but the question still is about the cost. And it's going to cost me $1,000 one heparin dosage versus $2 heparin dosage, whichever one I choose. I may want to choose the chemical one because it's more expensive, fine. But my insurance company will not let me use a chemical heparin. They may, like insurance company may still force me to use animal heparin because animal heparin is still cheap. So in order for you to overcome that, you have to reduce the cost of that, and also to be able to scale up that. Alternative would be stimulating anticoagulants endogenously, because we do make heparin in our body. We do make heparan in our body. So if we can stimulate endogenously, that would be another approach. And that's one of the approaches that our lab is started out with by using xanocytes, which we'll talk more, I think, towards the end. And so then the heparan sulfate derives as an anticoagulant. It can also be used, many different places and have many different roles to play. And it has been shown to play a role in tumor progression because the tumor cell basically is a normal cell. Now it became a tumorous because it has uncontrolled growth. That's the only difference. The tumor cell is not a foreign cell, it's our own self cell. But now it has uncontrolled growth behavior to keep multiplying, keep dividing, keep growing. And that's why it became a tumor, okay? And it causes its own problem. So it has been shown that heparan sulfate, plays a role in many stages of tumor progression. For example, not in carcinogenicity, it is not shown, but at least in tumor growth, it has been shown to play a role, because any cells to grow, they need a growth factor interactions. So HS-FGF interactions and is required for the cell growth And again, heparan sulfate plays a role in FGF binding and hence uncontrolled growth. And also it has been shown that in the tumor cells, they secrete number of enzymes which supposed not to be secreted. Normal cells don't secrete certain enzymes. Whereas tumor cell secretes certain enzyme like heparinase, which cleaves the heparan sulfate, not heparin ligase, but heparinase, which is a hydrolytic enzyme which cleaves the heparan sulfate into smaller fragments. And the small fragments then can bind to VEGF and VEGF receptor, and that can trigger the angiogenesis. So now tumor cells, as keep growing, they need the nutrients. So to get the nutrients, they need to draw blood vessels towards them. To draw the blood vessels, what they do there, they cleave the HS into small fragments that can bind to VEGF and VEGF receptors that trigger the tumor growth, because now we have blood vessels growing towards them to get the nutrients. And also by cleaving HS chains, and also they also produce matrix metalloproteases, and they start chewing the extracellular matrix proteins, and heparinase cleaves the heparan sulfate. Now the tumor cell has a HS chain which anchor to the fibronectin and other adhesive proteins and extracellular matrix. But now the extracellular matrix protein is getting cleaved by specific tumor cell specific matrix metalloproteases, and also HS, heparan sulfate, also getting cleaved. Now they're attached to each other. Now the protein getting cleaved and HS getting cleaved. Now they no longer anchor to each other. They're no longer adhesive. Now the tumor cell became less adhesive. It dislodged from the primary site. Now it's becoming a non-adhesive cell. Start rolling in the vasculature. Start rolling and rolling and rolling. And then by then you start making HS again, and then it can start anchored some other place. Then it causes secondary growth, and that's we call metastasis. And all these places, again, HS has been shown to play a role in growth, angiogenesis, and dislodge and lodging in another place, metastasis, and also invasion. In all those places, HS and also chondroitin sulfate in some other tumors has been shown to play a role. And no one yet has exploited HS-based drugs to treat the tumor yet, and except that a company, which company I don't know, that make angiostatin, like the VEGF antibodies to just block the angiogenesis, therefore you can have some beneficial effects. But not HS has been used yet as a drug to treat the cancer. A number of enzymes are upregulated or downregulated, which are summarized in this slide. And H-sulfone and sulfur, the one that Krishna was asking. And in some cancers they're upregulated, one of the isoforms. In some form they're downregulated, and it's not quite clear. But what is clear that heparinase is always upregulated in many different cancers. And also one of the key biosynthetic enzyme is 3-OST2, and which has been shown to be in some cancers silenced. X means silenced. By some other groups they say that it's upregulated. But nonetheless, in many different cancers, 3-OST2 seems to be silenced or slightly upregulated. So we don't understand the consequences of the silencing and so forth. But what has been shown by one group recently, that the 3-OST modification seems to bind to one of the chemokines, I forget, and that has a role in the pro-inflammation. Not inflammation directly, that triggers a pro-inflammatory signaling pathway. So by having 3-OST silenced, so 3-O-sulfation suppresses inflammation. By silencing 3-OST, now it cannot bind to certain factors, now it triggers the pro-inflammatory pathway. So it also been shown inflammation and cancer is linked. Continuous inflammation leads eventually to the cancer. That also been proposed and been proven by many other groups. So the cancer doesn't happen overnight. It happens because of continuous inflammation somewhere happens, and that triggers the cancer eventually. So before cancer, there's inflammation. Before inflammation, there's a pro-inflammatory stage that triggers inflammation eventually. It seems like 3-O-sulfation plays a role in regulating the inflammation by silencing 3-OST, and now the pro-inflammatory protein activated. They're turned on, by turning on, that triggers inflammation that eventually results in causing the tumor. But yet no one has exploited this knowledge to see whether by turning on 3-OST enzyme, by using 3-OST2 specific enzyme, can you shut down the tumor? We don't know. But maybe this could be a cause, or could be effect, we don't know. If it's the effect, then we can regulate as an anti-tumor agent, but if it's only regulating the inflammation that causes the cancer, then we cannot use this knowledge to treat cancer. But nevertheless, in tumor cells you can see the aberrant HS structures produced. And the heparinase enzyme that I just talked about, which can cleave heparan sulfate into smaller fragments, and therefore they can bind and regulate coagulation factors, adhesive molecules, and so forth, and therefore they have different roles to play. And what happens now in the normal cells, and there's so many enzymes involved in making HS, and so many enzymes involved in breaking this molecule, and there's a fine balance between biosynthesis and metabolisms. And so the enzyme which makes the long backbone of the HS is called EXT enzyme or polymerase enzyme. Mutation in that enzyme results in not able to make a long HS backbone. You make a very short backbone or no backbone. Oftentimes you make a short backbone. Instead of having 50 repeating disaccharides, you may only have a 10 or 20 repeating disaccharide units. And as a result of that mutations in this enzyme, it causes dwarfisms in humans, as you can see. And so then there's the mutations in the core protein glycans, which talked about glycan in one of the very first slides. So you have a glycan, and they have two GAG chains attached to it. And when the attachment site, the serine, serine amino acid, is mutated, what happens? You cannot transfer xylose to serine because serine is becoming glycine or alanine, something. So basically you cannot transfer xylose to that amino acid. That means that if you cannot transfer xylose, the entire GAG chain in one place is missing. You still make a glycan 3, instead of having two chains, you only have one chain. And that causes a syndrome in humans called Simpson-Gallagher-Behana syndrome, where the people have uncontrolled growth. And they're 7 feet, 8 feet, 9 feet, which is fantastic if you're only a basketball player. And but still it's very painful to be a 9 feet tall person. You can't fly, you can't take a car, okay, you can't go to the bus, you can't bicycle, and you can do hiking. A lot of things, okay, can go wrong. And you can't have a nice dress any place. You have to make a custom made. It's very expensive. And so except it's good to be like a basketball player, that's all. And then that charm and fame goes away once you get out of college, okay? And so what happens, because I know most basketball players, college players, they're all very poor now, and that's what I found, majority of them, unfortunately. And so what it tells you that a defect in the HS biosynthetic pathway have either undergrowth or overgrowth. So for normal growth requires all of these enzymes expressed in specific amount, in specific places, in specific time, and so that you have a normal growth. This tells you about how HSPG can dramatically alter the growth in humans. This is about the biosynthetic enzymes. Also they will be shown again by this giant Burnfield by overexpressing Proteoglycan, in particular syndican, in hypothalamus, because hypothalamus is the one that controls our feeding behavior, how much you want to eat, okay? You can keep eating even though you're not hungry, you can keep eating because you're addicted to certain food, because hypothalamus affects our feeding behavior aspects of that. By overexpressing a syndican in hypothalamus region, the mouse doesn't stop eating. It keeps eating and eating, it becoming big, giant, okay, you can see that here. And the wild type mouse is looking at you, see that? And this one is not looking at you because it just has become big, so. And so hypothalamus, when you overexpress heparan sulfate Proteoglycan, it has uncontrolled feeding behavior, keep eating without realizing that it has to stop eating, because once it's filling, you stop eating because there's a feedback mechanisms by which you will stop it. But here you don't stop it, as a result of that it becomes overgrown. Also in humans, it has been shown by a syndrome called Kalman syndrome. There's 6-sulfotransferase, which is the 6-sulfation of glucosamine residue. When this enzyme is absent, two things happens. There are hypogonadisms, sexual organs not well developed, and also these people miss olfactory bulb. That means that they cannot smell at all. And surprisingly, these people can live until age 50-55, and then the number of other coagulation problem happens. But until age 50-55, they are fine. But afterwards, number of cardiac problems and coagulation problems, they die. By age 50-55, they die. But the key thing is that they suffer from hypogonadisms, and also they have the olfactory bulb is missing. So all this slide summarizes defect in the HS biosynthetic pathways and how it leads to diseases. And so I think we should take a break. Terry's here, otherwise he'll break me. And so then we'll talk about how the metabolic enzymes involved and in regulating the metabolisms, and the absence of those enzymes can trigger its own consequences in humans. Okay.
Mhm.